Everyone, I think you're doing fine. So, in this lecture, I'm going to talk about some basic laboratory techniques by which we can isolate microorganisms from different samples. Well, actually, I will mainly talk about the basic laboratory techniques which we use to isolate bacteria from different samples. Different samples means like environmental samples, say soil sample, or pathological sample, or clinical sample, such as, for example, uh, throat swab, nasal swab, uh, stool, urine, blood. Okay, so first I'll talk about these basic laboratory techniques. Then I'll talk about briefly about the vaccination and chemotherapy. Okay, so okay, before going to the techniques, when we deal with microorganisms or play around with microorganisms, we need to have clear idea about some definitions. Okay, so these are actually important for us, for biologists, to have clear idea about these definitions. Okay, like, so first let's start with the species. Okay, so we all know what species is. Like, for example, we are human, like say you, I, we all are human. In what basis we are saying that we belong to the same species? Okay, we are human species, but how we can say we belong to the same species? This is also true for microorganisms. Like, for example, bacteria. There are a different type of bacteria. Say E. coli. Okay, E. coli is one species of bacteria. Okay, Vibrio cholerae, another species of bacteria. So, in what basis we say that these organisms belong to this species? Okay, so basically, a species refers to a group of organisms having same genetic content, morphology, metabolic pattern, and are able to reproduce. Okay, so that means we say, like you and I, why we belong to the same species? Because our genetic content, that is the genes or genetic material, we have similar genetic materials, our morphology is similar, metabolic pattern is similar, and also we are able to reproduce. Okay, so if the organisms, they have similarity in these things, then we can say they belong to the same species. Like all the E. coli, they should have similar this kind of things. All the Vibrio cholerae bacteria, they should have similar this kind of things. Like all the donkey, all the cow, all the goat, right? So the same species, all the members of the same species, they should have some similarities. Okay, similarities in these things. Okay, genetic content, morphology, metabolic pattern, and they are able to reproduce. Okay. Okay, there is one scientist, T.D. Brock. He is a very famous microbiologist. Okay, he defined uh, microbial species in a different way. Okay, so he defined microbial species as organisms who share 100% similarity in DNA sequence. Okay, for example, all the Bacillus cereus, they have 100% similarity in DNA. All the Escherichia coli, which is also called E. coli, have same have 100% similarity in their DNA sequence. Okay, okay. Although T.D. Brock defined this way, but actually, in reality, even all the members, actually two members of the same species, they don't actually have 100% similarity in their DNA sequence. For example, you and I, okay, we both are human, we belong to the same species, but still we don't have 100% similarity in our DNA. Okay, then microbiological culture. Okay, I have said that. Okay, in this lecture I'm going to talk about some basic techniques by which isolate we isolate bacteria. Okay, so basically what we do, what these things see, what this thing is called. Okay, this is called actually culturing the bacteria. When we grow bacteria in the laboratory, okay, we have a special like plate and a special some we use some special chemicals by which we grow the bacteria. These things is called culturing the bacteria, and that is called microbiological culture. When you grow the microorganisms in some in a plate, we call it microbiological culture. Okay, so what microbiological culture means? A microbiological culture means the growth of a defined population of microbes under defined set of parameters. For example, say a microbiological sample have been collected from an environmental, clinical or pathological origin. Okay, so after collecting these samples, we can grow them in a specific medium under a specific parameters. What parameters? Say specific temperature, humidity, pressure for a specific time. Okay, and this is called a culture. Okay, actually, it's different species. Even like if you think about the bacteria, different bacterial species they sometimes need different environmental parameters. I mean, some bacteria may prefer 37 degrees Celsius. Some bacteria may prefer 25 degrees Celsius. Even some bacteria may prefer 90 degrees Celsius. Okay, and different bacteria they I mean, different bacteria means different species of bacteria. They may prefer different level of humidity, different level of pressure, different level of pH. They may have different nutritional requirement. Okay, so that's why the definition is microbiological culture is like this: the growth of a defined population of microbes under defined set of parameters. Right. So actually, when we culture the bacteria, when we grow the bacteria in the laboratory, I mean, we can choose that what bacteria we want to grow. I mean, like we can choose, we can select that several species will grow in our culture, or we can choose that okay, we will grow only particular bacteria, such as we will grow only E. coli, we will grow only Bacillus cereus. Okay, so okay, so what kind of parameters we will set? It's completely depend on our intention. Okay, so if we want to grow many different species in our culture, the parameters will be different. Okay, but if you want to grow some very selective bacteria in the culture, then the parameters will have to be very selective for that particular bacteria. Okay. Okay, so now, I mean, I said that when we culture bacteria, okay, we have to provide them, so they provide them something. Basically, no, every organisms they need food. So why we eat food? Because food is, I mean, from the food we get all the nutrients that we need for our growth. So it's also true for bacteria. Bacteria are microorganisms. So for the growth of bacteria, bacteria also require nutrition, right? So that thing is called culture media. Okay, so basically culture media is the food for all microorganisms. Okay, or you can say culture media is the food for organisms. So we can define culture media in this way: culture media is the physical and nutritional substance on which microorganism is grown, and the culture media contains sources of different nutrients, like it contains carbohydrates, proteins, pH to support the growth of microorganisms. Okay, culture media can be of three different types: solidified agar, liquid broth, semi-solid media. Actually, I mean, in a different way, I can say like this. I mean, if you want to grow bacteria, you need to give them food. That is, you need to give them culture media. So you can give this culture media in a different way. I mean, you can give them very solid food, the solidified agar. Solidified agar are solid. You can give them solidified agar. You can give them food in a liquid. I mean, in liquid form. It is called liquid broth. Or you can make the food like not fully solid, not fully liquid, something in between, like a semi-solid media. Okay, so basically, culture media have these three different forms: solidified agar, liquid broth. The liquid one is, I mean, when it is solidified, it's called agar. Okay, because agar is a powder which you have to put there to make it solid. So solidified agar, a liquid broth. Broth means liquid. Okay, whenever it is liquid, we call it broth. Okay, semi-solid one. Okay, and this photo actually it shows different culture media. Okay, so I mean this left one, this so these are some solid agar. Okay, solid culture media, solid agar, and this right one is a liquid culture media. It is called broth. Okay, broth means liquid. So now, I mean, when we culture the bacteria, we can culture it in a different way. It's not like different way. It means like suppose you can culture the bacteria. I mean, suppose you take a soil sample and then you want to grow the bacteria of that soil sample in the media. So you can culture them in a way that after culturing you will get different species. Okay, you will get at least more than one species, or you can get only one species. Okay, you can culture in this way. Either, I mean, you can culture in a way that there will be only one species, or there will be more than one species. So if there is, okay, if a culture has more than one species, it is called mixed culture. Mixed, okay, more than one species. Okay, so for example, if you collect sample from the soil contains many different types of microorganisms, such as it contains virus, bacteria, fungi, algae. Okay, so if you just, okay, so this is called mixed culture. But if you culture in a way that only one particular type of bacteria will grow, so for example only E. coli will grow, it will be called pure culture. Okay, so that means solid culture, they actually contain more than one species in it, and pure culture means there is only one species, only one type of microorganisms. Okay, so that's why, so it can be, I mean, you can define the pure culture in this way. When a culture has cells from only one species, it is called a pure culture. Okay, so for example, if you make culture soil sample. Okay, so if a soil sample is cultured and then diluted and processed to isolate every single species, then we have pure cultures of many different bacteria, such as for example Aspergillus fumigatus, Bacillus subtilis, Bacillus cereus, etc. Okay, actually, there is way. Okay, I'm coming to that. So I'm saying that we isolate bacteria. So what actually isolation means? So if I ask you what microbiological isolation means, I mean, I think in this lecture several times I said that isolation of I mean, we want to suppose you want to isolate bacteria from soil. So what this isolation means? What this microbiological isolation means? Okay. So microbiological isolations refers to the method of selecting cells from only one species to grow in one specific culture. Okay, so when you say microbiological isolation, it means to that method, okay, in which method we can select cells from only one species. That means only one species will grow there, okay, in one specific culture. Okay, so this means microbiological isolation. Okay, so now let's go back to the soil thing. So you have a soil sample, and the soil sample is a mixed culture, right? In the soil sample contains different type of microorganisms like the virus, bacteria, fungi, algae. Okay, so now your goal is you want to isolate only one species. Okay, that means you want to isolate a pure culture from a mixed culture. Okay, so how we can do this? Okay, so well, we can do this in two basic methods. One is by serial dilation to extinction, and another is growing on a specific media. Okay, that specific media can be a solid media, semi-solid media, or a liquid media. Okay, so for this part of lecture 3, that's all. In the next part, I'll talk about these methods. Okay, the serial dilation to extinction method are going on a specific media. So by, I mean, these two methods, we can isolate a pure culture. That means a single species from a mixed culture. Okay, so a mixed culture means where there are several different species. Okay, okay. So see you in the next part. Thank you.